I'd like to welcome all of you at the first talk at Biological Seminar. We have new program for this summer term, but just several of them. So we have a look of them. It was finished this Sunday, so it's totally fresh. First, my pleasure is to introduce the next speaker, which is in front of you. It's Jaroslav Fleger. So next week we will have Jaroslav Fleger, something about rabbits, evolutionary psychology, and Facebook. So if you are more interested in this topic, come next week or ask Jaroslav after this talk. However, it's my huge pleasure to welcome here today Jordan Okie from Arizona State University, who is guest by David Stork, but David is ill, so I have a pleasure to introduce you. David doesn't have this pleasure. I am very happy to have this opportunity, thanks to David. So maybe some of you remember that we already had the title From Microbes to Monsters. It was told by Jim Brown about more or less the same or similar topic, and that's why there is the number 2.0 advanced version of the same talk by another speaker. Welcome, Jordan, and we are looking forward to your talk.
Thank you. Thank you, everyone, for coming. So, ecological and evolutionary dynamics have organized life into these different levels of organization. Starting with the origin of life, metabolic networks got integrated into a cell, and through endosymbiosis, there's the evolution of eukaryotes, where these endosymbionts evolved to cooperate with the host cell as mitochondria, and then cells cooperate in tightly integrated fashions to form multicellular organisms. Some of these organisms, as they get larger, end up with transportation networks to take up resources from the environment and distribute them to the different cells. And here we see a variety of other ways that ecological and evolutionary dynamics have organized life, including lots of plants and animals which are in close association with other microbes living on and within them. So to try and understand life, we need to try and understand principles that apply across levels of organization, and how the biological form and structure might change with these different levels of organization. And that's going to be kind of the central focus of today's talk. And in particular, I'll focus on these transitions here, which are called major evolutionary transitions in individuality. So these transitions were really important in the history of life on Earth. They transformed biogeochemical cycles in the biosphere, and they allowed for the evolution of a lot of novel morphologies and physiological structures and new niches. Each transition required the cooperation between individuals to form new individuals at a higher level unit of organization. So that's why it's called a transition in individuality. Metabolism and size played an important role in these transitions. So there were changes in form and function associated with the transitions, and those changes were also solutions to constraints that were limiting metabolism and that led to new constraints. So here are some examples of that. We can see that if we compare two different cells of the same size, say a prokaryote and a eukaryotic cell, but the eukaryotic cell, its respiratory membranes involved in ATP synthesis are internalized on mitochondria, and by internalizing those surfaces, it can actually have a higher surface area for respiration than a prokaryote of the same size. So that's an example of a metabolic change in metabolic design that was of benefit to the eukaryote. Likewise, multicellularity has an effect on the uptake of resources. If we compare two different organisms of the same size, one that's a single-celled organism and one that's a multicellular organism, the multicellular organism will have less problems with acquiring and distributing resources from the external environment to the different cells and organelles within its body, because it can have these smaller cells then, and it can have cavities that allow resources to have closer contact with the cells. Okay, and so to understand the interaction between size and evolution, we can take advantage of the study of allometric scaling relations. These can really offer powerful insights into the fundamental processes that constrain and regulate biological structure and function. Kleiber's law is a law that has been proposed by biologists, suggesting that metabolic rate, the amount of energy consumed by an organism per unit time, should scale with body mass as a 3/4 power law. So here's B is metabolic rate, M is body mass, and the exponent, according to Kleiber's law, is 3/4 across all of life, going from unicells all the way up to plants and animals. And so to look at Kleiber's law, biologists will log transform both variables so that then there's a linear relationship indicates a power function where the slope is the exponent in this power law. There are two aspects that are significant about Kleiber's law. So one is that the exponent is less than 1. So that's what I'm calling sublinear scaling. And that means with increasing size, metabolic rate is increasing less than proportionally, so that a larger organism has a lower metabolic rate per unit mass than a smaller organism. And this sublinear scaling is often attributed to some combination of constraints related to the collection of resources from the environment, surface areas of organisms because they have to take up resources across their surfaces, and within organism distribution of resources and waste products, so the transportation networks can lead to sublinear scaling. And also, with larger sizes, often organisms need to allocate more tissue to biomechanical support, such as skeletons, endoskeletons, and that can then lead to less tissue available for metabolically active reactions that would lead to a linear metabolic scaling. The significance of the 3/4 value in particular suggests that there might be universal mechanisms that underlie metabolic scaling across all of life. And it contradicts an old law that biologists call the body surface law, where in its sort of naive interpretation, that is that metabolism, metabolic rate, should just be proportional to the external surface area of an organism, and all else being equal, if we compare organism sizes here, their surface area scales to the 2/3 power area, to the 2/3 power volume and body mass, and so then we'd expect metabolic rate to scale to the 2/3 power body mass. So the fact that biologists are observing this 3/4 scaling suggests that maybe this body surface law isn't as very relevant to understanding scaling. In addition, there's some other reasons for studying allometric scaling. So a lot of rates in biology and time, such as generation time and biomass production rates, growth rates, rates of uptake of different nutrients. They're often affected by metabolic scaling and metabolic rates. And a variety of models at other levels of organization, like in ecology, assume different allometric scaling relationships and then use that to build models to predict flows of carbon in an ecosystem. So another example is population growth rate, and which is a measure of fitness, and that is fueled by metabolism. So it's often thought that the maximum population growth rate should just be proportional to mass-specific metabolic rate, and thus, according to Kleiber's law, with exponential quarters, then our max should decrease with increasing mass. Another quantity of interest to biologists is efficiency. So energetic efficiency of biomass production, that's the ratio of the growth rate to mass-specific metabolic rate. So it tells you something about how much mass a population or organism can grow given a certain amount of energy that it consumes. So these scaling relationships in Kleiber's law are really interesting, but in working on this several years ago, I found it really surprising that there was this Kleiber's law because there are these really profound physiological differences between prokaryotes and eukaryotes and multicellular organisms. And here, this is just a kind of a summary of these differences. And so just to refresh your memory, so prokaryotes, the respiratory complexes are located on the cell membranes, the genomes are relatively small, and there's less active transport and cytoskeletal structure within the cell. In single-celled eukaryotes, the respiratory complexes are instead on the inner membranes, so they're internalized on the mitochondria within the cells. And in multicellular eukaryotes, then, as the organisms get larger, they have to have much more elaborate transportation networks, that they have to allocate a lot of biomass or tissue towards developing. To study these scaling relations then across these transitions, I worked closely with John DeLong, a colleague of mine, and we put together in a variety of organisms. And we tried to address some of the previous problems with the data in unicellular organisms. So we made the observation that biologists, in accepting Kleiber's law across all of life, had not actually very carefully put together the data in microbes. They hadn't accounted for variation in physiological states and temperatures. They had often used individual measurements rather than taking species averages that would allow you to compare across species. And so we tried to account for these kinds of methodological and statistical issues. And so this is just an example of some of the organisms and some of the species that we got data for. And we focused on heterotrophic organisms in order to avoid introducing another confounding variable that might affect the scaling relationships. So we actually found that there were shifts in metabolic scaling across the transitions. So one single fit is misleading, and the relationship between metabolic rate and body mass is improved by taking into consideration these levels of organization, these different groups. So here in this figure, this is showing metabolic rate versus body mass, and here these are prokaryotes. And the black lines show the active field measurements, whereas the blue line shows inactive measurements, starving measurements, where the organisms aren't being fed very much. But in both cases, there's a very steep scaling between metabolic rate and body mass, where the slope is close to 2. It's 1.9 and 1.7. So it's a very steep relationship, which means that with increasing body mass in prokaryotes, metabolic rate is increasing even more, disproportionately so, such that the largest bacteria have higher metabolic rates per unit body mass than the smaller bacteria. In protists, instead, the relationship is linear. So it's super linear in bacteria, and now in protists it's linear. And that means that the mass-specific metabolic rate is invariant of body mass. Kleiber's law and the sublinear scaling only emerges in animals. So there are these categorically fundamentally different scaling relationships across these different transitions. We also looked at energetic efficiency of production. That's R max divided by mass-specific metabolic rate, and found that it decreases across the transitions, and used a model to evaluate the role of the transitions in body mass in influencing efficiency. And so here, this is the efficiency of production, and what we found is that for the most part, it's not changing very much within a group, although there's some indication that maybe it decreases a little bit within animals. But the major changes are across groups. Okay, so what are the implications of some of these observations? So metabolic scaling is not governed by one single design principle, and instead we need to think a little bit more in an integrative fashion about how the combination of constraints might be affecting scaling across these transitions. Each transition incorporates additional structures, and these structures require energy for maintenance, and so that leads to a decrease in efficiency. So, inspired by the data, I've been developing theory to try and understand these scaling relationships. And the core of the theory is that the major evolutionary transitions allowed escape from external surface area and internal distribution network constraints that would normally impose sublinear scaling in larger sizes. So here is a conceptual diagram summarizing the theory. So if we want to understand metabolic scaling, we have to understand the constraints on metabolic rate at a given body mass. And there's a constraint related to the surface area of the organism and its ability to distribute resources from external environment to within the organism's body. So that's sort of like can be thought of as a supply constraint. It's very upstream on the chain of metabolic reactions involved in metabolism. And because of surface area and distribution constraints tend to lead to sublinear scaling, and so it's a shallower scaling relationship. In contrast, the rate of downstream metabolic reactions, the catabolic reactions, those can scale much more steeply because they're just involved in the reactions located on the mitochondria or within the cell's cytoplasm. And so the metabolic scaling then is going to be the blue line, which is wherever one of these supply or the sort of demand-like constraint is affecting the scaling relationship. And thus, because of these two, the supply and demand constraints, that can lead to a shift from steep scaling to shallow scaling within a group of organisms. Now let's see how this then applies when we think about the major evolutionary transitions. So here's one group of organisms here, such as unicellular eukaryotes, and we found in the data that they exhibited very steep linear scaling for the data range that we had. Now that's not to say that the gigantic cells, which are not in our data set, sublinear scaling might not happen. And what happens during an evolutionary transition? Well, a transition allows an innovation in metabolic design, as I showed earlier in the introduction. And so that shifts the supply constraint up. And so in turn, that allows this new evolutionary group to continue linear metabolic scaling until it hits, again, the supply constraint that imposes sublinear scaling. Now, there's still some key questions related to this. So, where does the groups fall within these scaling constraints? So, do they tend to be more in the linear part or the sublinear part? What determines the exact slopes of the scaling relationships? What determines the range of overlap in the sizes of the two groups? So one group might still be able to evolve to be smaller along the linear path. Okay, so now we can apply this theory to the different transitions. And so I'm going to go in more detail talking about each group, but first let me provide a little bit kind of a summary of the general idea of applying this to the transitions. So in prokaryotes, the scaling we saw was superlinear, so very steep. But at some point, cell surface and distribution constraints have to come into play, leading to sublinear scaling. When that happens, the endosymbiosis of the respiratory membranes through the internalization of mitochondria can allow linear scaling in protists until, again, the sublinear constraints come into play. And that's when then the evolution of multicellularity can again allow linear scaling. But again, it's temporary. At larger sizes, the difficulties with obtaining resources and distributing them within the body are going to lead to sublinear scaling. But I haven't yet addressed this question of, well, why is it so steep, the scaling in prokaryotes? The hypothesis or theory I'm building is that it's related to the number of metabolic pathways and genome size, which I'll now talk about. So, empirically, genome size increases with cell size. And our hypothesis is that the larger genomes then can confer more power. And there's a couple of different ways this can happen, but the general idea is that so you have more coding genes, that leads to a greater diversity of enzymes possible in a cell, and so then a more complicated biochemical or metabolic network. And such larger biochemical networks can then use a greater diversity of different substrates as energy sources. And different diversity of metabolic pathways can be used to process different substrates. And they can also use these nutrients at a greater rate or derive more energy per unit substrate. So let's look at the data a little bit to see how much of these assumptions seem to hold. First of all, the genome size, both in terms of the number of genes as well as the total base pairs, does increase with cell mass in prokaryotes. Additionally, in several different researchers, this is just one example, have shown that the number of reactions in the metabolic network increases with the number of genes, which isn't surprising, right? In more genes, you can code for more enzymes, and so there can be a greater number of reactions. And the number of metabolic pathways in the metabolic network, so that's the number of different ways that any substrate can be processed through the metabolic network, and that also increases with the number of reactions. And so by having more metabolic pathways, that increases the versatility of the network, because it can then use substrates in different ways to try and maximize the efficiency of substrate use. So if we put together all these patterns, what would that say about metabolic scaling in prokaryotes? This is sort of a proof of concept, so it's not meant to be completely rigorous, but it's to try and get a handle on, does it really seem possible that this explanation can explain superlinear metabolic scaling in prokaryotes. So the basic idea is that the metabolic rate of a cell is related to the summation of all the reaction rates of all the different reactions in the network, the metabolic pathways. So the R is the reaction rate of the metabolic pathways. And then delta G, that's the energy yield of a reaction. So that's the amount of energy that a reaction provides. And so if we take the summation of all those different pathway reaction rates and energy yields, that tells you what the metabolic rate of the cell is. And so that's basically the number of metabolic pathways times the reaction rates and times the energy yield of those pathways. So empirically, I've shown genome increases with mass, the number of individual reactions increases with genome size, and the number of metabolic pathways also increases very steeply with the number of reactions. So that suggests that the number of metabolic pathways increases roughly with this exponent of 0.7 or 0.8 with cell mass. Now if we combine that with expectations about how the total reaction rate of the pathway can increase with mass, which we expect is somehow related to either the surface area of the organism or the total volume of the organism. So the exponent should be between 2/3 or 1. We can then combine this information about the metabolic network scaling and the reaction rate scaling, and that would predict that the metabolic rate in prokaryote should scale with some exponent roughly between 1.4 and 1.7. Now I haven't said anything about this energy yield and how that would change with mass. So this is assuming the energy yield is invariant with mass. If the energy yields of the pathways are also increasing, that could also make the scaling steeper. So this kind of analysis then is suggestive that this hypothesis may be the explanation for superlinear metabolic scaling in prokaryotes. Okay, so now let's talk about metabolic scaling in heterotrophic protists. Now that these protists have internalized their respiratory membranes, and so then respiration rate can just increase the number of mitochondria until cell surface and distribution constraints come into play, leading to sublinear scaling. So biologists have shown that respiration rate indeed tends to be proportional to the total surface area of the mitochondria inner membranes. And in order to maximize scaling then, a cell would want to increase the number of mitochondria while keeping the volume, the size of the mitochondria invariant, because then that allows it to really just increase its surface area of mitochondria linearly with cell size. So this is then the predictions of the scaling relationships. And this just kind of illustrates, again, this idea. If you have two different eukaryotes of different sizes, by increasing the number of mitochondria, we can just increase the surface area linearly here. So this linear scaling is a consequence of the internalization of these metabolic surfaces by endosymbiosis. Then I put together some data to try and evaluate this theory. And so this was by both assembling data from the literature as well as using different techniques to analyze micrographs of cells and mitochondria. And the theory seems to hold. So the number of mitochondria, is increasing linearly with cell volume, where the exponent is 1.04. Whereas the size of the mitochondria are invariant to cell volume. There's no relationship. So that suggests that in these heterotrophic protists that are exhibiting linear scaling, the supply of resources to mitochondria is not constraining their metabolisms. And of course, what about really gigantic unicellular eukaryotes? Maybe at that point there would be constraints related to the uptake of resources. And so that's something that would be interested in putting more data together to try and understand whether or not sublinear scaling occurs in the really larger cells. Let's see. Okay, so now I'd like to talk about scaling in plants and animals. And again, the same general idea applies. So the scaling at first can be just related to the surface area of mitochondria, and so scale linearly with number of cells until these other constraints related to transportation networks and surfaces come into play. So indeed, actually, near linear scaling in tiny plants and animals has been observed, such as in this, these sorts of aquatic animals here, like a hydra. And there's a variety of theoretical support that transportation networks, as they become more extensive, will end up leading to sublinear scaling in larger organisms, such as a human. Here's a human with a very developed cardiovascular system for distributing resources to the cells. If we look at high quality data that people have put together, there's indication of this scaling shift. So this shows whole plant respiration rate versus whole plant mass, and it's a little bit steeper here in the smaller plants, and then the scaling gets shallower. So these researchers here, they fitted like a smoothing regression to the data. It's not very obvious though, right? But if one applies a linear line here, it fits the data quite well, and then at some point the data is deviating from the linear relationship and showing a scaling exponent that's closer to 0.8. So there does seem to be an indication of the shift in the constraints that are affecting metabolic scaling. Okay, so I've talked about these different organisms, prokaryotes, single-celled eukaryotes, and multi-cell eukaryotes. But what about eusocial colonies? So like ants or termites. These colonies can be very tightly integrated in terms of their uptake of resources and processing, their metabolism. So we can think about these colonies as organisms or superorganisms and examine how their colony metabolic rates scale with their colony mass. So again, here we might expect the scaling relationships to scale linearly with the number of individuals or the number of mitochondria, but at some point surface constraints come into play or transportation networks come into play. This is some rough data that look at the metabolic rate of both unitary insects in red and then in black colonies. And as you can imagine, it's a little bit harder to measure the metabolic rates of colonies. So the data are a little bit rougher and there's not as many measurements. These researchers, they just fit straight lines to the data, and the fits are okay, but there is some indication maybe of some residual structure here with the points not, the line here is not falling directly through the center of these points here. So perhaps it is steeper and then gets shallower at some point, although more work has to be done to really try and rigorously test this idea. And if we look at colonies, it seems possible that also there's a steeper scaling and then shallower. But since we only have 2 points here, this is only suggestive, and a lot more work needs to be done. Okay, so I've shown some data here that indicate that the constraints on biological design and metabolism have played a really important role in these major evolutionary transitions. And these constraints have shifted at different scales, and so that then leads to mixed scaling relationships. There isn't just one universal 3/4 Kleiber's law, but rather the scalings can shift within a group as well as across groups of organisms. This kind of, I think that this work really emphasizes the importance of the internalization of surfaces in these evolutionary transitions. it shows that because of endosymbiosis, this naive expectation that the surface area for metabolic processes should just scale with volume to the 2/3 exponent is misleading. That because like through the endosymbiosis of mitochondria, we can actually see linear scaling of the respiratory membranes. There's also a variety of other strategies for altering the surface areas and the scaling of these surface areas, both in evolutionary and in ecological timescales. This is just a figure emphasizing the variety of different strategies available to organisms and showing kind of how they play an important role in physiological ecology and evolution at multiple levels of organization. So I've talked about internalization here, such as with mitochondria, but there's two other sort of basic fundamental categories of surface area scaling strategies. Organisms can have really convoluted fractal-like surfaces, or they can change their shape with changes in size. So they can have smooth surfaces but just change the shape, such as getting really long or really flat, and that will alter the scaling of the surface area with body mass. Or they can get more hollow with increasing size. And we see these sorts of strategies both in unicellular and in multicellular organisms. Okay, so I've talked about looking at scaling of the metabolic rate of organisms with organism mass, but we can also apply this approach to looking at the scaling of organelles or cells or other units that are at lower levels of organization and see how they scale with their sizes. So for example, here, within a cell, the mitochondria might be of different sizes, and so they will exhibit some scaling relationship between their metabolic rate and their size. And within a multicellular eukaryote, there are cells of different sizes. So what's the scaling relationship between cell size and between metabolic rate and cell size, and how did these transitions in levels of organization, such as endosymbiosis or the evolution of multicellularity, affect metabolic scaling of these lower level units? To look at this, we should consider that the metabolic rate of a cell reflects both the number of organelles as well as the size of their organelles. And so here, the number of organelles times the metabolic rate of an organelle is going to be related to the cell's metabolic rate, right? Because if most of the respiration occurs on the mitochondria, then it's the cell's respiration rate is going to be related to the number and the size of those mitochondria. And so if we put these different relationships together, so number times metabolic rate of organelles is proportional to the metabolic rate of the cell, and substituting in the scaling relationships, we can see that organisms can change the number, the size, or the scaling of their organelles in order to match increasing metabolic demands of cells. And so there are three questions that arise from this, and that is we can consider, how does the number and size of organelles change to meet increasing metabolism of larger cells? And did endosymbiotic evolution of the prokaryote ancestors of mitochondria and chloroplasts alter their metabolic scaling? And does multicellularity affect organelle and cell metabolic scaling? So I'm going to talk about efforts to address each one of these questions now. So first, looking at the number and size of organelles. If we think in terms of the big picture of the evolution of life, right? So there was this community of prokaryotes, and some cells are large and some are small, and at some point through this endosymbiotic event, the evolution of a eukaryotic cell occurred. And as these eukaryotic cells get larger, they can either increase the number of mitochondria within them, or the size of these mitochondria. And so this is showing a strategy of cells increasing the number of mitochondria, and here it's showing them just increasing the size of their mitochondria in order to match the increasing metabolic demands of a large cell. Now, within multicellularity, we can ask the same question. So multicellularity allowed the diversification of all these different cell types, and some cells within a plant or animal are really large and some are really small. Do the small cells have fewer mitochondria? Or do they have the same number of mitochondria but just smaller mitochondria? So those are the two potential extreme scaling strategies. Of course, organisms could also follow a strategy in between that. And so, I put together data to investigate whether or not, what strategies organisms are using and whether or not these strategies are affected by the major evolutionary transitions, so the evolution of multicellularity. And so here is a summary of the results. On the left here we have plots of the number of chloroplasts and number of mitochondria versus cell size in unicellular and multicellular organisms. And the analyses indicated that the scaling relationships weren't significantly affected by multicellularity. So the scaling of number of mitochondria is the same roughly in both unicellular and multicellular eukaryotes. Likewise, if we look at the total volume of mitochondria within a cell, we observe the same kinds of scaling relationships, where the scaling was not affected by multicellularity. However, there are differences between trophic lifestyles. So you'll notice that the number of mitochondria here is scaling linearly with cell volume, whereas the number of chloroplasts scales with sublinearly, so much more shallow scaling relationship. And this relationship here too is more shallow. So in the number of mitochondria with cell volume in phototrophs, although it's close to linear, it's more shallow than the scaling relationship in heterotrophs, and that was a significant difference. So there's an effect of trophic lifestyle, but not of multicellularity on the scaling of the number of organelles with cell volume. And I also found that organelles weren't changing in size in a consistent fashion with changes in cell volume. So the cells increase the number of organelles with increasing cell size, but don't alter in any consistent fashion the size of their organelles. Okay, so next I'm going to tell you about how we can then use this approach to try and infer something about how the metabolic scaling of organelles in cells is changing across these transitions. So consider again this relationship between the number, size of organelles and the size of cells. But since the number of organelles and the size of organelles weren't strongly correlated, we can rearrange this equation and use a general linear model or multiple regression to estimate what these scaling exponents are, which are the metabolic scaling exponents for cells and organelles. And so that's just by looking at how does the number of organelles change as a function of both organelle size as well as cell size. And then we can use the general linear model to compare between evolutionary groups. Okay, so what did I find? First of all, the metabolic scaling of organelles changed as a consequence of endosymbiosis. So mitochondria exhibit linear scaling of respiration rate, whereas, as I showed you earlier, prokaryotes, which were the ancestors of mitochondria, exhibit superlinear scaling. So endosymbiosis fundamentally altered the scaling properties of these cells that then evolved into mitochondria. Likewise, chloroplasts and cyanobacteria were affected by this process of endosymbiosis. In cyanobacteria, so in phototrophic bacteria, the scaling relationship is linear or superlinear. There's still more work in the literature that needs to be done on this. But it's definitely different from sublinear, which is what I was estimated with the data for chloroplasts. So this process endosymbiosis altered the scaling relationships of both the mitochondria and the chloroplasts. In contrast, the evolution of multicellularity did not affect the metabolic scaling of respiration or photosynthesis of cells. So this, I observed sublinear scaling of photosynthesis in chloroplasts and cells, both within a unicellular organism, such as this euglena, as well as within a multicellular plant. And likewise, the scaling wasn't, for respiration rate, wasn't affected by multicellularity. Although you can see here that there's a difference, again, in the trophic lifestyles. So respiration rate scales linearly in mitochondrial cells, both within a cell and both in unicellular organisms as well as in cells of multicellular organisms. So there's a steeper scaling relationships for heterotrophic organisms or organelles than there is for phototrophic organelles or organisms. Let me take a little bit of time to talk a little bit more about this difference between the scaling in heterotrophic and phototrophic organisms. So there's this package effect. It's known as a package effect in the literature, which is that the idea that as cells get larger, light is going to get reflected and diffused throughout the cytoplasm more. And so essentially there's more shading and reflection of light with increasing cell size. And so that leads to this surface kind of constraint on photosynthesis that algae ecologists and physiologists expect would lead to sublinear scaling at even fairly small sizes in phytoplankton. Whereas in protists and cells within animals, they can attain this linear scaling by simply because the oxygen is not necessarily limiting their scaling relationships at those scales. Okay, so in conclusion, to sum up all these relationships, I showed you that Kleiber's law doesn't apply across all of life, so there's no universal three-quarter scaling in unicellular organisms. Instead, the scaling shifts both across groups, but also it can change within a group. And so we saw that there's steep scaling in prokaryotes, in heterotrophic prokaryotes, and then there's linear scaling in heterotrophic protists. And in multicellular organisms, there's initially steep scaling, but it quickly changes to sublinear scaling because of transportation network constraints. And possibly there's the same kind of thing in superorganisms, so eusocial colonies. Another question is, what is the scaling relationship for multicellular prokaryotes? So there are a few multicellular prokaryotes. They're not as common and as well known, but that may be because we haven't been looking for them. And it would be interesting to try and understand what the scaling relationships are in these multicellular prokaryotes, as well as in consortia in microbes. So prokaryotes are not living alone usually. They live in tightly packed communities. And in some cases, they're so closely integrated that they'll even form like a sphere with external cells on the, sort of forming a shell on the outside of the sphere, having one type of metabolism and then a completely different species on the inside of the sphere. And these two different species then completely depend upon each other in an integrated fashion. And so I think it'd be very interesting to look at the scaling relationships in these consortia to try and understand then the degree to which these principles apply across these different levels of organization. So this is just kind of showing again how life has been organized into these different levels of organization, and I think there's a lot of interesting opportunities for investigating scaling and also just the metabolism and ecologies of organisms and how they differ between these different levels of organization, going from single-celled organisms to multicellular organisms and communities that are structured in different ways. That, for example, it'd be very interesting to understand how different sizes of cells affect the scaling of endosymbionts within unicellular organisms. So these results kind of can help us elucidate then the role of metabolism and scaling in the major evolutionary transitions, and also point to some interesting future avenues for research. Okay, so I'd like to acknowledge my close collaborators on the work that I presented today: Jim Brown, John DeLong, Melanie Moses, Richard Sibley, Felisa Smith, Val Smith, and David Storch. Also my funding from NASA, ASU, and the Howard Hughes Medical Institutes. So thanks so much for listening, and be happy to take any